We'll start.
Well, good afternoon, everyone. I'd like to welcome you to today's edition of the HNRCA Monday Seminar Series. I'm Dr. Roger Fielding, the Associate Director here at the HNRCA. It's my pleasure to introduce today's speaker, Dr. David Drew, from Massachusetts General Hospital and Harvard Medical School. Dr. Drew is an instructor in medicine and Director of Biobanking for the Clinical and Translational Epidemiology Unit at Massachusetts General Hospital. He also has appointments in the Division of Gastroenterology, the Department of Medicine at MGH as well. The title of his presentation is Diet and the Gut Microbiome, Lessons Learned Towards Precision Prevention from the Predict Studies. Just a little bit of background about Dr. Drew. He received his Bachelor of Science in Biochemistry and Biophysics from Rensselaer Polytechnic Institute and received his Ph.D. in Biomedical Science with a concentration in Genetics and Developmental Biology from the University of Connecticut. We are really pleased that Dr. Drew could join us today for the seminar, and we're really looking forward to his presentation. So I'll turn this over to you, Dr. Drew. And remember, folks, when we get to the question and answer period, please submit your questions through the Q&A, and then I will read them and Dr. Drew will respond. Welcome, Dr. Drew.
Thank you, Dr. Fielding, for the kind introduction and the invitation to present our work with you today. I just want to confirm that you can see the slides out all right now. All right, great, thank you. So yeah, we're very excited to share our work with the Predict studies with you today. And I know this is a good audience that will have multiple kinds of interest in the work that we've done here. I'll just quickly disclose that Zoe Global Limited did provide research funding for the overall Predict studies, though we were not drawing salary from these studies, but the studies themselves were supported by Zoe Global Limited. I'll also provide a disclaimer that I'm not a nutritional epidemiologist by training. As you heard Dr. Fielding say, I'm a basic scientist, translational researcher at MGH and do the director of our biobanking. And I was very excited to do the Predict studies with a large team of investigators. But my background actually is in chemo prevention of colorectal cancer. And this is a really good example for where precision prevention and what we think about every day when we think about precision prevention and where we see the field going. And I'll just start here mainly because I know this is the Center on Aging, and while there's a nutritional link and an aging link, both of these things have strong links with colon cancer as well. So as we think about aspirin chemo prevention of colorectal cancer, we also think about age and nutrition a lot. And this is where our interests intersected and allowed us to get involved in this area of precision nutrition, which I think also applies broadly to human health and disease. I mean, a good example of this is we tend to think clinically with these one-size-fits-all approaches to prevention. And this is largely due to the fact that studies for prevention of disease, be it either nutritional epidemiology-based interventions, dietary interventions, or medicinal interventions, are based on an average individual. So in 2015, there was a landmark decision from the United States Preventative Services Task Force that the evidence had finally accumulated to a point that aspirin could be recommended for the prevention of colorectal cancer in certain individuals. This tapered with age, but in 2018, a randomized clinical trial, and as evidence of how this balance can quickly shift from one side of recommendation to another side of don't recommend this, is that the Esprit study that was run in Australia and in the United States demonstrated a signal for potentially increased cancer-related deaths, particularly from colorectal cancer, despite not having an increase in incidence. And there's many hypotheses for why this aberrant finding may have occurred within elderly individuals, but it had a drastic impact on how the field felt about aspirin chemoprevention, with just last week, the USPSTF announcing draft recommendations, reversing their landmark decision in 2016, and now suggesting that aspirin shouldn't be used for primary prevention of when considering colorectal cancer risk. So we're used to dealing with this idea of precision medicine. And when we think about this in terms of age, we realize that we can take broad scale recommendations based on someone's cardiovascular disease risk, their life expectancy risk, their risk for cancer, their risk for adverse events from aspirin intervention. And we've proposed a model that integrates molecular endpoints that allow us to identify responders, or those who are most likely to benefit from an intervention, versus those who will not derive much benefit or may actually be at an increased risk. So how does this apply to diet? Well, dietary guidelines and nutritional advice also take on this one-size-fits-all approach. And the evidence is constantly evolving. It's confusing for patients. It's confusing for individuals on what you should be eating, what you shouldn't be eating. Every given week, something changes. And what we know as a field is that that's not our endpoint goal, is to give these blanket recommendations and hope that they all work. We really are trying to tailor these to individuals, but it leads to lack of consensus. And this gets picked up by individuals. So when we look even among the leading experts in the field of nutritional epidemiology and dietary interventions, you would expect that maybe there should be perfect agreement. But when we actually survey them, we see that outside of the extremes of dietary interventions, we see a very heterogeneous recommendation panel. You see some people recommending some foods that are actually strongly felt to be bad among certain groups. And it could be that people just pick their favorite thing and that's what they study and that's what they know. And then everything outside of that kind of becomes a broad generalization. But ultimately, we also think underpinning this is that we're complicated as human beings and food is complicated. And so when we think about why food is complicated, you have all of these chemicals and nutrients in each food, and separate from like what we do with aspirin, which is a single molecular agent, you still can experience different kinds of interactions. So nutrient-nutrient interactions, the complexities of those foods, how those foods are processed, what they're eaten with, the food matrix that they present on. And it also leads to differences in even the same foods and the way that we consume them having significantly different effects on our metabolism, such that this study, which showed that whole almonds actually had a lower kilocalorie per serving effect than ground almonds. So the difference in the matrix can have completely different effects on each individual. Throw in the fact that humans are also complicated. And a lot of our advice is based on population-based averages of the average human being and how many of us are actually average. So if we consider that same example from what we just saw with the almonds, on average, a whole almond's metabolized energy was around 4.6 kilocalories, but the range was from 2.3 kilocalories to 6 kilocalories for each individual. So if you consider that in terms of the average serving size, you now can see big swings in the difference of metabolized energy from these same food groups. And then when you throw on top of this that nutritional studies typically show a mean response, and that's what we generate our advice from, but then we look at individual responses to food and we see that that's all over the chart. And this really highlights and underscored the need for personalization. But we also have the power now in 2021 to expand beyond traditional randomized controlled trials, which, while having high quality and high precision, are limited in terms of their number and the outcomes they can study. And then typical epidemiologic studies, which are more numerous, may not have the same kind of quality and precision necessary to identify individual responses to food. So big data provides this opportunity and new ways to capture this data to understand individual responses to food. So in 2018, we started conversations to launch the Predict studies. This is now a very large ongoing program with the goal of measuring postprandial responses to food. It's a collaboration between investigators at King's College of London, our institution at Mass General Hospital, Stanford University, as well as Tufts and the HNRCA. This is really a pairing of nutrition academia as well as tech companies. So Zoe Global and other biotech companies are partners in this approach to be able to generate high fidelity data and deep molecular insights into each individual. So I'll spend some time going over these studies because I think this is most relevant to this audience and people will be most interested in what we've done and how we implemented this and how we tried to build this program. So Predict is now in its third phase of a study lifetime. In Predict 1, we took 1,100 individuals from the United Kingdom and the US. It was a clinic and home phase day, and I'll get into the details of what the study actually entailed eventually, but the overall goal was to look at postprandial responses. We leveraged an existing cohort in the Twins United Kingdom study to be able to bring in high fidelity research participants, right? People who were used to being poked and prodded and filling out questionnaires and supplementing that with citizen scientists from the general population who could come in and help us by participating in our research program. 1,000 of these participants were in the UK, and then we had an individual validation set in the US, which is where I came in, as well as Andy Chan at MGH. We led the US study aspects of Predict 1. There were several sub-studies of Predict 1, including Predict Carbs, Predict 1 Plus, and Predict Cardio. I'm not going to go into those details today for the sake of time. However, I do want to point out that we have since expanded and completed Predict 2, which is essentially a mirroring of what we did in Predict 1, except in only at home phase and only United States individuals were participating to try to have a comparable data set to the Predict 1 studies and also try to expand, validate, and refine the prediction models that were coming out of our precision nutrition approaches from Predict 1. And then in Predict 3, this is actually kind of like the Apple Watch studies. This is built on Zoe's consumer product where people will buy the Zoe kit, they will get personalized insights into their nutrition, and then we as investigators will be able to get additional data, additional endpoints from those individuals who consent to participating in additional research studies as a part of them buying the commercial product. So I'll also point out that this work has been published. So while I'll go through and provide some high overarching views of what we've accomplished in this space, if you're looking for the real nitty-gritty details just for the sake of time, these two papers are going to be at the core of what I talk about today. I welcome you to go look at them. Many of you are probably already familiar with them, but I will go through and do kind of a broad overview of what we found in this space. So in Predict 1, our goal was to use genetic, metabolomic, and metagenomic and meal context information to predict individuals' metabolic responses to food. And really our aims were to determine how much variability there was between people, including twins, because from the Twins UK study, we had the ability to understand the effects of genetics and identical among a twin study, but also expanding this to a larger group of individuals coming off the street. We were able to try to understand what explained those differences across the individuals. And then Zoe and we as an academic group really wanted to understand, can we use this information to predict individual responses using deep data approaches? So overall, our goal is to integrate individual response with interrelated multi-directional pathways to power our deep data collection and do robust biobanking. And this, where as the director of biobanking at the CTU at MGH, I had a really strong interest because we are constantly thinking about what we can do to more deeply phenotype individuals beyond just basic anthropometrics or lifestyle data to try to move away from these one-size-fits-all approaches and have a better understanding of preventative or interception medicine. So when we think about how we respond to food, especially in the context of this study, we were using postprandial responses. Now, in typical feeding studies, you may measure or a fasting study, you may measure someone's response after a single meal intervention and try to understand how they respond and predict outcomes as a result of that. However, in real life, we go on multiple excursions throughout the day. You have breakfast, snacks, lunch, and dinner, which ultimately we basically live in a postprandial state all day after our initial meal in the morning. So with Predict One, we wanted to make sure we were able to study both of these factors and understand, is our single meal response is key to understanding inter-individual differences, or do we also need to understand how people react throughout the day to their postprandial response? And why do these peaks matter? Well, if we think about triacylglycerol and glucose and insulin levels, we realize these excursions go up as a result of food, but they also may stay elevated throughout the day. And this can have impact on a number of different downstream functions beyond from inflammation to normal biology to hunger and appetite. And all of these things together can increase our risk for cardiovascular disease, metabolic disease, or weight gain. And really, we need to understand how each individual person can respond differently to the same foods. So I'll spend some time on this slide, as this is the overall design of Predict One, and it can be a little complicated. So the power of Predict One was to combine a classic feeding study inside a clinical environment where we can do tightly controlled blood draws postprandially, specific interventions where we can watch patients, we can ensure that the metabolic challenges that we're giving them are administered the same way. We can do detailed and fresh sample collection. So individuals were invited to come to either King's College of London, St. Guy's Hospital in London, or in the US, and I can speak most about this, is to MGH, to the Translational Clinical Research Center, where they were seen for a full day in our clinics there. Individuals came in fasted overnight. They collected a stool sample at home that they brought in with them. We went through a series of anthropometry, DEXA, waist hip measurements, BMI calculations. We collected a urine sample. We collected a saliva sample. They filled out questionnaires, the standard food frequency questionnaires that we see in epidemiologic data, as well as detailed lifestyle and medical history questionnaires for us. They were connected to blood pressure and heart rate monitors throughout the day. But ultimately, the power of this was that we were able to do a feeding study and then do timed blood draws throughout a clinic day for a full day. And you can see here in the schematic where we collected blood and at what time points to better understand both the response to breakfast and then later a lunch intervention, and then send them home and follow them for a 14-day period at home. Now, we captured their dietary diary via a mobile phone app that was developed by Zoe Global. This phone app allowed them to take pictures of their food, scan barcodes, get very detailed data for us that allows exactly to know what they were eating. They were provided scales, so we had weighed dietary records. And we were also able, through the app, to be able to provide in-study support. So our clinical research coordinators and nutritionists could provide advice on how to log foods, make sure that things were clearly logged. If any patient had any question, they had 24/7 support to be able to get this, to get the data quality as high as humanly possible, despite this being at home and not being done in the clinic. They were sent home with standardized meals that included varied test breakfasts and lunches that were given in the form of breakfast. We also gave, or of muffins. We also gave a standard OGTT test. Participants collected blood spot cards for the first 4 days while they were at home. We used these for tag and C-peptide assays later. They were fitted with a continuous glucose monitor while they were in the clinic that they wore for the entirety of the duration at home. They wore a physical activity monitor on their wrist, which also allowed us to monitor their sleep. And then they provided a stool sample at the end of the study that they mailed back to us with their blood spot cards, their CGM, so that we could get the data and their physical activity monitors. And again, this was a main cohort of 1,000 individuals in the UK and 100 individuals in the US. Just to give you a sense of the scale of this study, we completed this study in about a year across the United Kingdom and 3 months in the US. It was a huge team effort to get that done. 32,000 muffins were consumed. 132,000 meals were logged that allowed us to capture not only the test meals that we intervened with, but also the free living meals that individuals were able to record within the app. We had over 2 million CGM glucose readings as this was being constantly monitored, and we had 28,000 tag readings from those blood spots. The demographics of the cohort is on the right. You can see the breakdown in terms of monozygotic and dizygotic twins, the non-twins in the UK cohort, and the dropout as well. Our cohort skewed female and to be middle-aged, around 45 years of age, around a normal BMI, but the higher end of BMI at 25. And these are just some of their fasting tag insulin glucose levels and cholesterol levels. The initial results really demonstrated the high significant variability between otherwise healthy individuals that we see for tag, glucose, and insulin. We also saw that within-person variability was lower than that inter-individual variability, which suggested that individual responses could be reliably predictable because they were repeatable, those differences in those individuals. So here we're looking at the 6-hour rise in tag and the, oops, sorry, I somehow got to the end of my presentation. Give me 1 second. Okay, we should be back, sorry. This is a 6-hour rise in tag as well as the area under the curve from 0 to 2 hours for glucose. And surprisingly, or maybe not surprisingly, depending on who you are, identical twins had very different responses. And what we see here is that genetics actually was not explaining most of the nutritional differences in terms of how individuals were responding to different foods. So that led us to ask the next obvious question: what is causing the variability in responses? And what we see is that blood-based markers as well as meal composition can be strong predictors of tag or glucose responses respectively. But as I'll get to further in the presentation, the microbiome came out as a strong signal in terms of predicting variability across individuals, which may explain why some shared environments may result in shared responses. But in terms of genetics having a strong impact on the response here, these other factors are what are driving those inter-individual differences that we see. In terms of were we able to accomplish our goal, what we found was that the machine learning approach that we had to predicting individual responses based on all of these different factors was very strong. We were able to measure this very strongly for glucose responses. This was validated within the US cohort as well, separately from the training set that was the UK cohort. And what is probably most encouraging from what we were able to derive from Predict1 is that because of the high resolution and high density measures that we collected, we were able to do deeper dives in terms of correlation to inflammatory markers, understanding the impact of hunger and energy intake, understanding lipoprotein remodeling, and also put this in context of individual risk factors. So this 6-hour increase in tag from their fasting values, we found was very strongly associated with intermediary cardiometabolic risk factors. And this led us to understanding that by doing deep phenotyping of individuals at baseline and healthy individuals, we may better understand baseline responses that can later change to dietary interventions that will ultimately have clinical outcomes that will be meaningful for patients. And then our cross-sectional interest here is also that our group is very interested in the how the gut microbiome interacts with different interventions, outcomes, how it mediates our health as another organ of our body, despite it being somewhat not what we considered a typical organ of our body. So just in terms of where the gut microbiome intersects with this area, we know that diet's a well-studied risk factor and a determinant of health and chronic conditions such as obesity and metabolic syndrome, all the way through cancer and cardiovascular disease. More and more evidence is pointing to alterations in the gut microbiome being a mediating factor for both diet's impact on chronic disease or health, but also as a direct determinant of health and disease. And several studies have linked the gut microbiome with food and metabolic health and trying to understand the complexities that this field faces. But surprisingly, the specific details of how the microbiome plays a role in obesity and cardiometabolic health have proven difficult to somewhat define reproducibly across these larger human populations. And it could be due to the fact that we all have our own ad libitum diets that are very different from one another. Being able to perform microbiome studies at scale can be challenging and costly, but also what we've learned is that individuals' microbiomes are highly personalized as well. And this persists over time, even with insults from intervention or other exogenous insults that may perturb the microbiome. So Predict 1 allowed us to do a very deep dive of metagenomics within stool samples provided from individuals at baseline from both the UK and the US cohort. So we were able to deeply sequence individuals' metagenomes and pair that with the high resolution data that we were collecting from the clinic day as well as the at-home phase of the study. For those of you who are not familiar with microbiome research is not that dissimilar from how we think about sequencing the human genome or doing other typical sequencing. Other than that, we take whatever microbiome samples we have, in this case, stool samples, we're able to then extract both host and microbial DNA from the bacterial cells and the host cells that are within those stool samples. We then sequence it per our standard sequencing next generation pipelines. And then we get this data set of all of these sequences and we put it through many computational workflows to decontaminate out host DNA and look specifically at microbial DNA, as well as be able to then map that back to the microbiome as a whole to understand not only who is there, but what is their genomic potential for what they can do, but also what they are potentially doing by inferring protein-protein interactions, pairing this with metabolomic data to better understand what they're producing. And then by standardizing this, as many of the team members on our team who focus on the metagenome and the gut microbiome as a whole have been working towards, is figuring out ways to standardize this across studies to more reliably use gut microbiome data, both for biomarker development, but also potentially to understand human health and disease much more consistently. So when we look at the alpha diversity or the taxonomic diversity of what is residing in our gut microbiome, how many different species there are, how many different types of species there are, we see that there are clinical factors and established biomarkers that weakly associate with some of these crude measures of the gut microbiome, meaning an overall view of the gut microbiome. So we see here that liver fat probability, as well as HDL, can be strong predictors of alpha diversity in terms of when we look at these among the subset of other clinical factors and other established biomarkers, but overall the correlations are relatively weak. So we needed to do a deeper dive to understand how the gut microbiome may be associated with all of these other individual factors. Why are our individual gut microbiomes potentially predictive? So when we do a machine learning regression and classification model using the gut microbiome features, we find that we can predict the intake of foods, nutrients, and patterns from individuals' microbiome signatures. And what we see, there are some stronger predictors and some weaker predictors. Coffee, for example, is a very strong predictor of who or what may be in your gut microbiome, but we also see that it can predict not only just foods, but different food groups, the nutrients that are within those foods, as well as overall dietary pattern, where you can see healthier diets may be more predictive overall of certain gut microbiome signatures. And I'll get into that a little more deeply in a moment. So the low-hanging fruit question is, what are the microbes that are associated with specific foods? And, as somewhat of a sanity check, we were able to see, certain bacteria, including like Bifidobacterium animalis, being strongly associated with yogurt consumption, for example. So classic potential microbiome and food associations we are able to pick up in our data. But overall, what we see is that there are two kind of distinct clusters that you have a health associated with a more healthy plant-based or a healthy animal-based diet on the right-hand side, showing certain enrichments or depletions of different microbial species, and then unhealthy dietary patterns that are being associated with a opposite effect for some of these species. And when you start to break down what the dietary constituents are, we see dark chocolate and jams can be associated with these health associated clusters potentially, whereas more processed foods like tomato ketchup, baked beans, and sauces are on the left-hand side of this diagram. Processed meats, including sausages and red meat, all the way on the left as well. Shellfish and oily fish and eggs, and those that may be high in fatty acids, fall somewhere in the middle. And all we really want to pull away from this is that not everything is necessarily good in a good class or bad in a bad class, but we do see that individual dietary patterns do tend to segregate individuals' microbiomes. And for those of you who are interested, if you like red wine falls in its own little subcluster here, pretty close to fish. So what about food patterns? Well, so in individual bugs, well, we can see certain enrichments for certain bugs according to certain food patterns. So healthy plant-based diets tend to have enrichment for this Firmicutes CAG95, whereas you see depletion of these bugs in the less healthy plant-based diets. Similarly, when we look at animal-based diets, we see different enrichments and different depletions for specific bugs. And really the take home here is it just reiterates what we saw before. Specific patterns, specific types of foods may be associated with specific types of bugs or specific patterns of microbiome signatures that we see in an individual. So we next looked at the effect of age and BMI and adjusted these things to understand whether or not there were tight correlations between certain bacterial species and dietary quality or other diet or health associated microbes among a curated list. And what we see is, again, there are certain differences in the bacteria that appear in clusters that appear according to food groups, the nutrients that are there, or the dietary patterns individuals follow. And what this really stresses is not only the importance of which foods you're eating, but the importance of the quality of the food, be it highly processed versus unprocessed food, the food source, be it plant or animal, or the food type, meaning that not all of some certain type of source are considered healthy or unhealthy, in order to understand both an individual's health, but also the microbiome ecology and what's happening within someone's gut microbiome. So in addition, we then look to see if there were associations between taxonomy and cardiovascular health. And what we see here is that visceral fat is actually a stronger predictor of what is in your microbiome than your BMI, for example. So again, suggesting biological mechanisms can underpin what is happening in the gut microbiome. And maybe we need to necessarily think about factors outside of standard lifestyle or anthropometric factors to be able to really understand individuals. So deeper molecular characterization, deeper clinical characterization of these individuals may provide better insights into an individual response to food or an individual factor related to the gut microbiome. So like with diet, healthy and unhealthy microbial signatures also segregate good and bad cardiometabolic markers. So again, and this suggests that the gut microbiome is not only mediating potential health outcomes, it's not only mediating the way we respond to foods, but it also can be a marker of what is happening biologically in each individual. So in summary, the gut microbiome is even more personalized than what we kind of consider as a person's self-biology. That diversity partially explains our personalized responses to food. The overall microbiome composition is strongly associated with dietary habits. We see that in multiple different signals in our data. And healthy, especially plant diversity in diets, is highly predictable from the gut microbiome. In fact, you can predict someone's dietary patterns from the constituents of their gut microbiome. These signatures that are associated with obesity are reproducible. They also appear to be linked with diet, and fasting and postprandial cardiometabolic markers are similarly linked to the gut microbiome. And in all, this really, for us, reinforces the idea that there's a strong reproducible microbiome signature that can give us some readout of overall cardiometabolic health and a healthy diet. So beyond Predict, our group obviously has a broad interest in the gut microbiome and health and disease. And Andy Chan, who I work very closely with at MGH, and our broader group has been examining similar effects for the gut microbiome and the protective association between different dietary patterns and cardiometabolic risk factors. So from the health professionals follow-up study, for those of you who are familiar with these studies, this is a large longitudinal study that's been carried out through the Channing Division of Network Medicine at the Harvard School of Public Health and Brigham and Women's University and Harvard Medical School. These individuals this is a group of men who have been followed since the late 80s. We get biennial food frequency questionnaires from them. It's updated every 4 years. We get detailed information on medication use and medical diagnoses, lifestyle. But Among a subset of men, we were able to collect, and healthy men, largely healthy men, they were free of cardiovascular disease or cancer. We collected stool samples at several time points several years ago. We were then able to pair this with longitudinal diet assessments, do deep metagenomic sequencing and metatranscriptomic sequencing of their stool samples, and also similarly pair this with cardiometabolic disease biomarkers. And this paper has also similarly been published, but what we found was there was a significant association with a low effect size of a Mediterranean diet with overall gut microbial composition, similar to what we were seeing with the Predict studies, again, but this is more of a dietary pattern with a specific gut microbiome composition. And what we also saw was that the gut microbiome can, in an individual's gut microbiome profile, can modify the potential protective association of Mediterranean diet with cardiometabolic disease risk downstream. And what was perhaps most interesting was there was a stronger pronounced effect, protective effect of those who had Prevotella copri. Those who did not carry Prevotella copri. And what this is an example of is as we go from a holistic view of the gut microbiome, trying to understand the microbiome networks and interactions that are resulting in differences in human health, there also can be candidate biomarker bugs that can come out of the gut microbiome that may help us molecularly profile, molecular phenotype individuals to understand their response to food or their likelihood to benefit from certain dietary interventions. And we've similarly seen links for fiber, the gut microbiome, and chronic inflammation within the same cohort. And this work that was led by Wenji Ma, who is on our team at the CTU at MGH, similarly looked to find that recent fiber intake was inversely correlated to BMI, as we might expect, but not correlated with age. And while the source of that dietary fiber was primarily from cereals, fruits, and vegetables, we saw that there wasn't large-scale differences in terms of the overall gut microbiome composition with fiber intake. However, when we look at individual microbial members, we see that they are associated with recent and long-term fiber intake and their CRP levels. And for us, this is another example of where you can potentially identify biomarkers for understanding downstream metabolic responses or inflammatory responses to food or their role in disease, and identifying a key mediating role for the gut microbiome. And an example here again is from P. copri, which has come out. We see that microbiome structure modifies that association between fiber intake and plasma CRP, such that there's a significant interaction. And trying to understand non-carriers versus carriers, this could be a way to help us understand, okay, this isn't David versus Steve. We're trying to understand, is this person carrying Prevotella copri or not? And what will that have? What impact will that have on their relationship with food and the downstream metabolic processes or the downstream inflammatory processes that may come from that? So in conclusion, Predict-1 represented the first diet microbiome study to identify both individual components of the microbiome and a gut microbial signature that was associated both with dietary intake and cardiometabolic health factors. These signatures that aggregate microbiome and dietary components can be used to understand health-associated and anti-health-associated clusters. And the gut microbiome seems to be an effect modifier in the association between diet and cardiometabolic disease risk. We provided an example of Prevotella copri, where that may explain someone's responsiveness to a specific dietary intervention, dietary pattern, and understanding what that could have on the downstream health effects for those individuals. And this really does open the door for using the microbiome as a way to get into personalized dietary approaches for disease prevention. So when we think about where we are and where we're going with precision nutrition, back our prior experiences with this field were that the individual was the core to how we could personalize their dietary intervention. An appreciation for the microbiome has been developed over that time, and how that may result in different responses to food. But we realize now, especially from the results that we're seeing from Predict, is it's far more complex, and we need a host of individual factors to truly predict how someone's going to respond to foods. And we're looking forward to the future from our ongoing studies, new technologies that will allow us deeper access to phenotyping, including those that we've already used for Predict1, but those that may expand beyond that. For example, we don't have a way to measure TAG other than using dry blood spot cards. We don't have an at-home blood collection device that allows us to do metabolomics, deep metabolomics, as a way to understand individual responses to food during a free living period. And these innovations will be necessary to continue to drive forward this area of research. And just to tie this back into where we started with cancer prevention, many groups are thinking about, where do you intervene with the gut microbiome, and how do you take advantage of understanding this relationship, this complex relationship between the gut microbiome and the host, to improve overall health? And can you also use it for disease intervention? So we summarized and reviewed a paper and that was looking at deriving chemopreventive metabolites from the diet, which has been classically very difficult to do when we think about cancer prevention, in terms of being able to get enough bioavailability or high enough concentrations that may actually result in prevention or recurrence of cancer in the future. But if we think about as the microbiome as a new drug target, as a way to intervene, right, as a new chassis by which you can deliver different agents or dietary derived metabolites to a host, you may be able to engineer the microbiome. There are many groups that are doing this to be able to improve prevention, act as biosensors to allow us to get signals and readouts of an individual's inflammatory response, a metabolic response. And I think there's a lot of promise in this area that we will see a lot of growth in over the next decade. And we've seen an explosion of this over the last few years as we get more and more metagenomic and deep molecular characterization of these individuals. So I'm grateful for the opportunity to present our work here, but obviously this is a huge effort and was the result of many teams working together. I'll highlight the team at King's College, which was led by Tim Spector and Sarah Berry, our colleagues at Zoe, Richard Davies, Jonathan Wolf, and George Hadjigeorgiou, and their analytic team, who did a lot of the machine learning approaches for understanding prediction of individual responses, as well as our team in the clinical and translational epidemiology unit at MGH, specifically Long Nguyen and Wenji Ma, who I presented some of their work around fiber and understanding the gut microbiome's role in Predict, as well as Nicholas Sagata's lab, which led our microbiome studies and the bioinformatics around that in the Predict1 studies, and as we go forward with Predict2. So with that, I'm happy to take any questions and hope we'll have a fruitful Q&A session.
Well, excellent. Thank you, Dr. Drew, for a very comprehensive seminar. There are some questions coming in already. I just want to ask a question. We got a question from our center director, Dr. Sarah Booth, who's not able to submit hers on the chat. So she asked me to read it. So this is from Dr. Booth. Given how critical it is to capture the inter-individual variability in response to specific foods and diet, yet as a general rule, we convey and report averages, do you believe that statistical analysis and presentation of data in peer-reviewed journals or publications, for example, should be modified to better capture the inter-individual variation?
Yeah, I think that's a really good question. I think what we're learning, not only from dietary studies, but as we think about precision prevention or precision medicine as a whole, is we do need better ways to present this data. Simplistically, it's easier to present averages or means and try to understand the bounds of which the normal distribution is there. But there's information in the extremes as well. And it can be, we do need better ways to both disseminate that information and make it relatable for lay audiences to be able to understand, what our findings mean for them individually. I'll give the classic example is the chemopreventive slide that I showed at the end with the microbiome, the engineered microbiome, came from a very basic science paper where they were looking at a metabolite from broccoli being potentially chemopreventive, from broccoli and other cruciferous vegetables. And the lay media quickly interpreted this as a broccoli flavored yogurt, because obviously bacteria were involved, could be the cure for cancer or chemopreventive for cancer in the future. And that's obviously not what we would hope for in terms of an interpretation as researchers and or clinicians. And I think it's hard to go from these broad generalizable outcomes that we typically put in peer-reviewed papers to then have individuals take away personalized messages from it without taking these rapid swings from these one-size-fits-all approaches. So thanks, Dr. Booth, for the question. I think it definitely has challenged the way that we should be presenting this data.
So we've got a few other questions coming in here. Let me start reading them for you. One is, I noticed that you did look at sex as a variable in some of the microbiome analysis, but there's another question here about, have you looked at or somehow adjusted results for menstrual phase in the women that you have studied?
Yeah, I believe that was menopausal status, I believe, was included as a covariate in the model. Again, the machine learning approaches do consider, you put in a lot of input and they will consider what is most relevant in terms of the variance that it describes in the model. I don't recall there being any specific signal for menopausal status or cycle at the time. But I would have to talk to the folks who did the analysis to get you a more detailed answer.
Okay. Another question, for a number of nutrients and bioactive compounds derived from foods, host metabolism plays a much greater role than gut microbial bioactivity. Do you think that this will limit our effort for beneficial gut microbiome manipulation against risk for chronic diseases such as cardiovascular disease and cancer?
I mean, I think that's a really good question. I would say I don't know. I think what we're still trying to understand is how much greater a role host metabolism plays than gut microbial bioactivity. I think our results point to the idea that it's not preconceived notions of how hosts interact with foods may need to be challenged such that we account for these other extenuating factors like the gut microbiome to understand how we can manipulate the gut microbiome for the risk of chronic diseases. So I think we're starting to tease that apart. I don't know that we have a strong answer to that, but what our data has shown is, well, the gut microbiome is not the strongest predictor of someone's individual response to foods. For certain metabolisms and certain metabolites, you do see a greater proportion of the variance explained by what is going on in their gut microbiome. So there is clearly a larger role there than we probably have given credit for in the past.
I mean, I suppose it depends on which nutrient and which metabolite.
Right. That's really what our results, I think, have shown. Right. For glucose, that wasn't necessarily the case. Right. And again, for glucose, you saw a greater contribution of genetics as well. Right. But whereas other environmental factors were more important for lipids and TAG. Right. So I do think it's again, the one size fits all approach, not only for the interventions, also applies to the biology. Right.
Exactly. Okay, so we're getting lots of questions. So this question is about the predictive model training. You mentioned that you trained your algorithm on the UK patient cohort. Someone just keeps moving. I'm sorry.
Not a problem.
My question screen keeps moving, and so I lose where the, wow, just. So if this is about the predictive model training. You mentioned that you train your algorithm on the UK patient cohort. Do you see this algorithm as appropriate to use on other demographically similar patient populations, or does it need to be retrained based on the beta diversity and functional profile of each specific cohort?
Yeah, that's a really great question. So, and that's what we're really trying to understand by expanding Predict 2 and Predict 3, right? So with Predict 1, it was primarily a UK, mostly white, mostly female, mostly middle-aged cohort. With Predict 2, we extended that to the US. And while our results showed that the performance in Predict 1 was good when we looked at the training set and then the validation set in the US, we obviously want to understand if that prediction model applies more broadly and is generalizable to a larger population, especially as we think about a more diverse population in terms of age, race, gender, other health outcomes. And so in order to answer that question and be fully confident that the prediction algorithms are translatable to larger populations, we do need big data. We need that data from that 10,000 person cohort to then make sure that we can refine this down to a better predictive algorithm. So big data folks and machine learning people will say more data the better in this case, right? To be able to move towards data prediction. But as far as what we can tell, it is relatively generalizable, at least in terms of demographically similar folks, as we saw with the validation set in the US versus the UK. For some metabolites, not everything has the same predictive capacity as we move beyond that.
Great. Here's a question. Not all fiber is fermented by gut bacteria. Have you considered dividing fiber intake into soluble versus insoluble, and then evaluating associations with outcomes?
Yeah, so that we have, and Wenji Ma, who led that work, is very interested in this question. We've also been thinking about this in terms of gut motility and the impacts of that on that. It's obviously a very important question depending on what outcome you're looking at. So yes, we have thought about it. I believe we break it down as best as we can from the FFQ data in HPFS as to soluble versus insoluble sources of fiber. I can't recall the exact outcome that they had right now, but if you look into that publication, you may be able to find a specific answer for your specific outcome of interest.
Okay, this is a question I think about the feeding studies that you presented. How many people ate prepared meals and for how long? What proportion of their intake was due to prepared meals? And then what was the adherence like to the prepared meals? And can you identify and use the patterns / microbiome to follow progress of age-related diseases? So maybe that's two questions, a little bit about the technical things about the feeding studies, and then using the patterns of microbiome to follow progress of disease.
Sure, I'll start with the first one, just logistically. So that it was a 14-day trial. The prepared meals were obviously on day one. Participants took prepared meals all the way through to day 12 of their at-home period. It was only breakfast every day during that period of time. 2 of those days were an OGTT breakfast, the fasting, and then an OGTT challenge. Several days they also had a lunch, but dinner every night was free living. And then most days lunch was free living. So it slightly tilts toward prepared meals, but actually that's not true. It slightly tilts towards free living meals because we didn't have lunches most days. So again, that would be dependent on the individual and how many meals they ate each day, but they were allowed to free live outside of, in the morning, they had very specific instructions as to how they should consume prepared meals, whether or not they were allowed to have coffee, all of those things. The adherence was high. Again, that was largely due to the fact that we had a lot of interaction. We could track people in real time based on their logging within the app. So if they didn't log in the app, our coordinators would reach out to them and ask them to put that in. And then the last question is interesting. I don't know if I have an answer to that. I think we could look at whether or not there are specific microbial patterns that track with progression of age-related diseases, but I don't have a good answer to that at the moment. But good question.
Okay, another question. Were you able to make predictions for postprandial glycemic responses from the metadata and microbiome, and how did those models compare with those generated by Aaron Siegel's group at the Weizmann Institute?
Yeah, so I think they're referring to, yeah, I know Aaron Siegel's work. We have been able to do that, and Nicola Sagata has worked with Aaron as well as via Curtis Huttenhauer in our group as well. I don't know the specific prediction they're asking, but what we found was the postprandial glycemic response for that specific like the cookie banana issue that they've raised in the past, that the glycemic index value of a specific food group does not necessarily predict the response on the other side of this. We see that similarly with our data in that individuals respond differently to the same foods, but we find that it's not just postprandial glycemic response that's driving that. So the metadata in the microbiome allows us a another deeper look into what is happening with those individual responses. But I don't know how far beyond it expands from what Aaron did.
Okay. Another question about a specific bacteria. Do you have any insight into the predictors of who is a P. copri carrier?
That's a good question. I don't think I have an answer to it right now, but it's something that we could definitely look into to understand that there are specific associations with those who are P. copri carriers. We get that question a lot from participants in Predict 1 because blastocytosis came out in our analysis as being tightly associated with lower BMI and lower visceral fat, and individuals wanted to know how they could get it, how they could get this into their microbiome. And I think that's kind of where we need to better understand what are the predictors of how you become a carrier and have carriage of some of these bugs, or how can we intervene such that you would be able to engraft these things in a healthy way for individuals. And that's kind of where the field, I think, in terms of microbiome engineering or microbiome targeting for prevention and interception will move towards.
Thank you. I might take the opportunity to just ask a very brief question. I noticed you have detailed measures of sort of physical activity that you're monitoring in your cohorts using probably wrist accelerometry, but I was just wondering, in the phenotyping measures, are you measuring any indices of cardiorespiratory fitness or aerobic capacity or muscle strength or muscle mass or size? Because I think there could be some very interesting associations in those areas as well.
We haven't, to my knowledge, done that. Well, we definitely have not done in the US cohort. I don't know that they have plans to do that as a part of Predict 3. It's possible that they will enroll sub-cohorts in for that. The best measure we have, muscle mass would be from the DEXA scans that we did, right? And some of that analysis is ongoing, how to use the DEXA data most fruitfully. Some of these endpoints just became unruly to study in terms of carrying it forward to the next few studies. When you know you're going to go to an at-home phase, you're not going to have a clinic day. So the challenges for Predict 2 were to still get blood draws that we could use for fasting measures in individuals. So we created a network with Quest centers to be able to get bloods collected on our behalf. So participants across the US could go to a Quest center, get the bloods drawn, but they obviously don't have access to come in and get a DEXA scan. However, we could then go back to targeted pieces of the population and try to get some of these additional measures like you're proposing to be able to look at those associations. But, in terms of scope, there's only so much you can do. We try to do a lot, but only so much.
But even something like grip strength is a very simple measure that could be embedded in even a home-based assessment. So, I mean, it's certainly possible.
Yeah.
But no, totally understand. I guess one final question. Did any of the cancer-associated bacteria like Fuso show up in your analyses?
Yes, I think you're asking about Fusobacterium nucleatum. Fuso is typically not seen highly prevalent in stool. And many of the analyses that you see when you don't do whole metagenomic sequences, you kind of have to targetedly look for it. It doesn't show up in our larger metagenomic analyses, like from the health professionals follow-up study in healthy individuals. It's that in particular tends to be more of a cancer-associated microbiome. A lot of it is measured in tissue, but we don't necessarily usually see it in the healthy population stool. Beyond that, I think there's still, there are other bugs that have been associated with cancer that we do see in different prevalences within individual stool microbiomes. But I think the linkage between strongly cancer-associated drivers and versus passengers in the cancer-associated microbiome and understanding where they come into an individual's microbiome in terms of their status of health or disease is still unknown. But I don't think Fuso popped up anywhere.
Okay, I think that is our last question, and we are just about at our time. So without further ado, I would like to again really thank Dr. Drew for an outstanding presentation. This was very comprehensive and a really nice segue into some of the initiatives that are hopefully going forward on personalized nutrition and the role of the gut microbiome in this area. So again, thank you very much, Dr. Drew, and thank you all for participating in today's seminar.
Thank you.
Thank you. That was spectacular.